Hello, I'm Norberto Peporine Lopes, full professor in organic chemistry, working at School of Pharmaceutical Science of Ribeirão Preto, University of São Paulo, Brazil. On 9th of November in 1989, I was in Berlin. Based on this information, you know that I was present when the wall going down. It was an amazing experience, difficult to explain the feeling, but it was like freedom in the air, something unbelievable. And when I returned to Brazil, I started my career working in Amazon forest. I spent like 10 years in a very deep Amazon forest, working with seasonal and circadian variations of secondary metabolites, small molecules, small compounds of plants. And also I was in contact with very remote tribes with no contact with non-native persons. That was a second amazing experience in my life. And together I really understand at that time that it's really important to preserve the forests, but also it's really important to preserve the people and the natives. I spent the second part of my career working a little bit with savannas, and later in the last 10 years I start to apply metabolomics strategies, which means metabolomics from the people is it's like if you compare with a code bar, when you have every single line together give you a price of the product. For example, what we try to do is have every single compound of an organism in one information about the species that you are analyzing. And we apply the initial strategies in single species, and later we try it to combine this analysis of plants, metabolomics of plants, insects, animals, at the same time the bush, to try to understand the dynamic of the forest. Which means if you have any comparison, like when you have a disease, when you are sick, what you lost, the equilibrium of the body. It's the same idea that we try to use, that you need to find the equilibrium, the dynamic of the chemistry in the landscapes to understand the dynamic of the forest. Our group, for example, report for the first time the occurrence of chemical barriers from the secondary metabolites working like sunscreen in the top of the leaves in savanna, or we also showed for the first time the luminescence on frogs that break one of the dogmas of the biology. We work with borderlines between forests like landscapes from savanna, nearby landscapes of Atlantic rainforest, and then the individuals or the species that from in the border zones can amplify the chemistry. And in this way we're allowed to propose several rules and to clarify some theories of the evolution in biology, of the evolution of secondary metabolites in evolution. And some of these data were used from people from the government to make decisions, at least to think about how preserve forests and locals and natives. And we also, when do this kind of approach, we can valorate the biodiversity. That's another word that some government understand. In our case, for example, we have already started like 5 startups in our labs with the students, and all these startups is based on ecological friendly services. And in some cases you have new products coming from new material from the nature prepared by the natives, and then you can make this environmental work well. And then in this way I think the most challenger from the next steps of this work is try to combine the metabolomics, like I explained first, this code bar, with proteomics, when you do the analysis of the total proteins, with the genomics that try to understand the genetic of the organism, and put all the players in the same box. In this way you'll be much more strong to define in the next years the dynamic of the forest. And I believe that this kind of research can be useful to help the nature. It's really sad when you see, like in Brazil, the forest burning. And I hope our research still contributed in this way. Thank you very much for your attention.